So when we talk about protein structure, we like to break it into 4 levels as a way to understand what is happening inside of this shape of the protein. And so as an overview, those 4 basic levels is first, the amino acid sequence. The secondary structure is a series of small shapes within the protein, like an alpha helix or a beta sheet. And these will fold in and become part of a 3-dimensional object that I call the blob, right? For many proteins, they're done at this point. And then the final level of structure, the 4th level of structure, is the quaternary structure. And that can happen when this needs to become part of a larger protein by interacting with other shapes. We're going to go through each one of these now individually. So the primary structure is just the amino acid sequence. This is determined by the genetic material, so the sequence in the DNA will lead to a sequence in the amino acids. Remember I said earlier that every protein is going to have an N-terminus and a carboxy terminus, and then there's the order of the amino acids. Secondary structure is when within that sequence we begin to take on some shapes. And so just for kind of how I simplify it a little, I'll draw it over here. So imagine this is my primary structure, is just a straight line, right? My secondary structure is when I start to see little shapes within that string. So I might have a little knot here, I might have a little helix here, I might have a pleated sheet, another coil. So there's little areas of shape within the string now. Those little areas of shape form because of hydrogen bonds in the backbone. So this is not involving the R groups, just the backbone, the carbons and nitrogens in the repeating part of the protein. And they will make hydrogen bonds with each other to take on these shapes. These shapes can be a helix called an alpha helix, or a sheet that goes up and down. Imagine like a paper that you've accordion folded. It's kind of the shape it takes on. That's called a beta pleated sheet. Or they can take on just a little coil shape, which is my kind of like knot that I drew in the string. And these are like, again, little mini areas of shape within the larger protein. But it still hasn't taken on a 3-dimensional shape for the whole protein. The tertiary or 3rd level of shape, and let me just go ahead and redraw this here. I've got my primary, my secondary, and then my tertiary is when this all folds up into a 3-dimensional object. And again, it's like the blob, right? But not the blob in a random sense, because this is now the final shape of the protein for many proteins. For some proteins, they're done. This is now a functional object that can go out into the cell and do its work. The tertiary structure is facilitated by interactions in the R groups. So not the backbone, but the R groups, or the variable part of the amino acid. These interactions can be hydrogen bonds between the R groups. They can be ionic bonds between charged R groups. Disulfide bridges are covalent bonds between sulfurs in the R groups. You can have hydrophobic interactions. And hydrophobic interactions is where nonpolar R groups have to be away from water. So the protein will have to fold up in such a way that R groups, which these are nonpolar R groups, they have to be folded up inside of here so that they can be away from water. The other type of interaction here is the van der Waals interactions. And van der Waals are very, very weak, even weaker than a hydrogen bond, and they are an attraction between any 2 atoms that are near each other. And again, it's a very, very weak interaction. That's why it can be between any 2 atoms. We're not talking charge or anything. Just any 2 atoms that are close enough to each other will feel a certain amount of attraction. So when this folds up, It's putting atoms close to each other, and that's going to help stabilize the tertiary structure. Quaternary structure. So again, let me do my little drawing here. Is when I have multiple three-dimensional blobs, if you will, that have to come together. So here's two different proteins going through this process. So primary, secondary, tertiary. This one's going to fold up. This one folds up into a different shape. And then the quaternary structure is when this and this have to find each other, and there's some sort of interaction between the two, and this is your final functional protein. So this one on its own is not functional yet. This one on its own is not functional. They're only functional once the two find each other and come together. So when this is the case, these are referred to as subunits. So this would be one subunit, this is the second subunit, and this is a multimeric protein. Multi means many, so there's two parts to this protein. These are examples from your text. So this is a multimeric protein. This is one part, here's another part. Many proteins are like this. Hemoglobin, we'll talk about again over the course of the semester, has 4 parts that have to come together. Collagen has 3 parts that have to come together. So many proteins do have to have quaternary structure. So again, just to review the 4 levels of protein structure, you have your amino acid sequence, you have secondary structure that puts alpha helices, beta pleated sheets, or random coils into the protein because of hydrogen bonds in the backbones. These structures will become part of a 3-dimensional structure where the protein folds in on itself completely. For some proteins, this is the final structure. In some cases, this will, in this case, if that's true, this has to become part of a larger multimeric protein, and only then would the protein be functional. I talked about these bonds before, but just to go through it in one more time, because this is important. You can have hydrogen bonds between the backbone and the R groups. So backbone was secondary structure, R groups was tertiary structure. You can have ionic bonds between R groups, and remember this would be your positive and negatively charged R groups. Hydrophobic effects are really important when you have non-polar R groups. They have to fold in away from water. That's what we see happening here. These are all non-polar R groups, and so they have to be folded up away from water. We see hydrogen bonds here between R groups, here between the backbone. And ionic bonds, this is a negatively charged R group, this is a positively charged R group. And I just want to point out to you, for example here, that notice that in the primary structure, these are actually not next to each other. They can only make the ionic bond when they're folded like this. So this curve here is allowing this R group to stick up from here, this one that comes out here, that's allowing them to get close enough to make an ionic bond. So the folding is really being held. It's almost like this is, after it bends, this is like glue that's holding that bend in place. Van der Waals attractions or van der Waals forces that I made mention of earlier is an attraction between any atom once they get close together. These are extremely, extremely weak. And then disulfide bridges, which we see an example of here, is a covalent bond between R groups that have sulfurs in them, so a very specific kind of interaction. So many of these interactions, like the van der Waals and the hydrogen bonds, are very weak. But as we said before, that when you have lots and lots of weak forces or bonds, they can still be very, very powerful. And one of the examples from nature is the gecko. And here's an image of a gecko, and here's a close-up of their feet. The gecko actually uses van der Waals forces, these very weak attractions, in order to cling to surfaces. So they don't have anything sticky on here. There's nothing sticky that allows them to stick. There's no suction. What happens is there's just millions and millions of van der Waals attractions between their feet, and a surface. And so you might wonder, well, how come I can't do that? Well, the reason why you can't do that is you don't have all these micro folds in your tissue. These micro folds create high surface area. That's all folded in. So even though it looks small, all these folds actually make it be really, really a large amount of atoms. So many more atoms from the folds that can interact with the surface. And so that creates a large, large amount of van der Waals attractions. And again, that's enough to bear the weight of the gecko. On a final note, these interactions that we've been talking about can also facilitate temporary interactions between proteins, so protein-protein interactions. This is not quaternary structure. Quaternary structure is permanent interactions between proteins. This is short-lived or reversible interactions between proteins. So this might be, and we've used the lock and key analogy before, these two proteins might need to come together to facilitate some sort of interaction, and they can be driven to come together by matching shape. So we have shape matches. So notice how this will fit here, this will fit here, this will fit here. But then they also, in addition to that, will use these same kinds of attractive forces. So the H bonds, the van der Waals, hydrophobic effects, anionic bonds can all help two proteins come together temporarily. All of those are weak interactions, and so this is a reversible process, which again in biology is often very, very useful. We want things to come together for a while and then separate.